leaves, roots, stems, branches, fruits, flowers, and seeds. All of these structures are produced by the plants, and then after their life cycle is over, they eventually die. Now, always remember, when we talk about the plant growth aspect of it, the plant growth starts with seed germination. When the seed germinates, the root is formed, the shoot is formed, and then we can say that the plant growth has been started. So, the first step in plant growth is seed germination. Okay? Seed germination, because after the seed germinates, this is the point wherein the roots are formed, and then like roots are formed below the ground surface and stem is formed above the ground surface. Any doubts up till now? Hello, am I audible? All right, so moving forward, let us talk about the developmental processes in plants, and they are controlled by two factors. Okay, see, the development that I said, that development takes place by growth and differentiation. Now, this development which takes place in the case of plants, it is controlled by two factors. The first factor is the internal factor, which is known as intrinsic. Intrinsic means from the inside. The second factor that controls the developmental process is the extrinsic factor, or you can say the external factor. So when the external and the internal factor, they have sort of like the direct effect, or they directly control the developmental processes which takes place in the case of plant. Got it? All right, so moving forward. Now, see, talking about the growth in the case of plants, so it is accompanied by metabolic processes. Now, from the previous classes, you might remember that metabolism contains anabolism and catabolism, right? Can somebody tell me what is anabolism and what is catabolism?
Anabolism is synthesizing molecules.
Hmm.
And catabolism is breaking down of molecules.
Okay, and do you remember in which of these processes is the energy released?
Catabolism.
And in which of the processes the energy is absorbed?
Anabolism.
Very good. So the metabolic processes, they happen in the plant as, sort of like accompanying the growth in the plant. And of course, the metabolism would contain both anabolism as well as the catabolism. Up till now, do you people have any doubts in the whole thing? All right.
No.
So, okay. Moving forward, now I told you that the growth in the case of plants is indeterminate. First of all, we can't determine the growth of plant, and secondly, it is unlimited. Now, why do you think so? Why do you think would be the reason for their growth to be indeterminate, or you can say unlimited? Why? We established the fact that, okay, it is unlimited in plants, limited in animals. But why is it unlimited? Hmm?
They get unlimited energy.
They are unlimited?
They get unlimited energy.
I can't hear you.
They get unlimited energy.
They get unlimited energy. All right. See, unlimited energy, it actually doesn't make sense though. Like, where do they get unlimited energy from?
From the sun.
No, 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 no. No. See, the reason why their growth is unlimited is because they have meristems present. See, the growth can be unlimited when the cell or whatever organ or anything that keeps on dividing, so it will keep on growing. So how do the plants do that? How do they keep on dividing? How do they keep on growing? So they do so because of the presence of meristem. Do you remember meristems?
Yeah.
Yeah, the meristematic tissues when we are talking about the tissues in the plants. So the meristems, they are present at many locations in the plant's body. So it means that meristems are always dividing. These are the cells, these are the dividing cells. So these meristems will cause the unlimited growth of the plants. Got it?
Yes.
This is the reason why, because of the presence of meristem,
Yes.
this is the reason why they can grow unlimited. Their growth is actually indeterminate. We cannot actually figure out as to when will they stop growing, will they stop growing or not. Got it? Now,
Yes.
the type of growth in the case of plants is known as open type. What is the meaning of that? Open type of growth, if you remember from anatomy. Hmm? Hello, am I audible?
Yes, ma'am.
Yes, I was asking what is the meaning of open type? What do they mean? What is open? Roof? Floor? Tell me. Open type, you remember when cambium was present? Open type? No?
Yeah. Yeah. Yeah.
Yeah, you remember your most hated chapter in the world? Yeah, so the type of growth in the case of plant is open type, but here the open refers to like anything that does not have an end to it. Okay? Now, the growth aspect of the plant has two growth types. The first one is the primary growth that occurs because of the meristem, more precisely because of the this thing, apical meristem and intercalary. Talking about the secondary growth, it occurs because of the lateral meristem, which contains vascular cambium, cork cambium, and all of these things. Okay, so the growth in the plant can take place by two ways. The first growth, that the primary growth, and then comes the secondary growth. Clear? All right.
Yes.
Okay, good. Now, moving forward, growth can be measured. Now, this is now see up till or like up till here, this thing. Whatever they have said is like, I don't know why they have said it. But okay, since they have said it, so I'll tell you. So, see, growth can be measured. We all know that, right? Like how we measure us humans, we we measure the growth in terms of feet, right? So we say someone is 5 feet, some inches, this feet, this inches. Same way, there are certain parameters that we take in terms of plants to measure their growth. So we either measure the growth in the case of plants by fresh weight, by dry weight, by length, volume, area, and the number of cells. See, growth does not only mean growing in size, growing in length. It can mean that, okay, the weight of the plant is increasing, okay, the volume is increasing, the surface area is increasing. All of these structure things can be considered as the growth in plant, not just, increasing in length. Like how us humans can grow like horizontally and vertically, isn't it?
Yes.
When we grow vertically, we grow in height. When we grow horizontally, what do we grow in?
Fat.
Yeah, we get fat, we get overweight,
Yeah.
or anything of that sort. So, but in the case of plants, there is fresh weight, there is dry weight. These are the parameters with which we will measure the growth in the case of plants. Now, coming at the cellular level, the way we can actually measure the growth at the cell level is when we'll see the increase in the protoplasm or increase in the cytoplasm. And when we see that, we can say that, okay, the growth has been taking place, the growth is there. Got it? The growth or the increase in the cytoplasm It is actually the, you can say, indicative part is to tell us that, okay, at cellular level, is the growth taking place? Yes, if the protoplasm is growing. No, if the protoplasm is not. Got it? Any doubts up till now? Sure?
Yeah.
All right. Okay. Another thing I forgot to ask you people to switch on your cameras. Switch on your cameras.
Can I do it after some time?
Sure. Okay. Well, you see, if you don't want to, it's fine. I was just saying because that's how you people roll. If you don't want to, it's fine also. Because the topic is not that thing that you require my facial expression. Up till now, is everything clear? It's nothing. It's boring only, isn't it?
Yeah, yeah.
Hmm. Now let us move forward and see the phases of growth in terms of the plants. Okay, so there are three phases of growth. The first one is meristematic phase, then there is elongation phase, and then there is maturation phase. Talking about the meristematic phase or the phase of cell division. This is like if you say meristematic phase, or you can also call it as the phase of cell division. Okay, now this is the first phase of growth. It is the first one, the primary most it is there. Okay, now here what happens in this meristematic phase, the meristematic cells would perform all sorts of divisions, and because of which the growth will be taking place. Now always remember the meristematic cells, these cells are capable of dividing. So these are the dividing cells. Okay, so if I say that they are the dividing cells, it means that they give rise to or they perform the function of cell division, isn't it?
Yes.
So remember that the meristematic phase or the phase of cell division gives rise to like the proper, you can say, the body of the plant, the various organs and everything, they divide because of the meristematic cells. One such example is the tip of the root and the tip of the shoot, meaning here apical meristem. You remember apical meristem? Apical meristem. Also, like, can you people tell me how many chapters are left in the whole syllabus to complete?
2.
Plant growth and development we're doing. Apart from this, what are the two chapters?
Plant kingdom and structural organization.
Okay.
Yeah.
Okay. All right. So now moving forward, see, the meristematic phase has all types of meristematic cells, and all these cells, they show the following features. So the cells which are present in the meristematic phase, they have these features. The first one is that they have dense protoplasm. A very dense cytoplasm is present. They have large nucleus. Okay, they have a huge nucleus. They have high respiration rate. High respiration rate means that they are respiring very quickly, very fastly. So that is why they have a high respiration rate. Then talking about the cell wall. See, the cell wall can be primary or it can be secondary in nature. But when we are talking about the cells of the meristematic phase, so here the cell wall is primary in nature. What does it mean? It means that cell wall, when it is primary, it is made up of cellulose. So that is why the cell wall is primary in nature. Also, the cell wall contains abundant plasmodesmatal connections. See, plasmodesmatal connections are the connections of plasmodesmata. Plasmodesmata. Now see, these are sort of like the connections between two cells. For example, if this is one cell and this is another cell, these two cells will be connected with some sort of connection, and this connection is the plasmodesmatal connection. This is like, why is it there? So it is there so that the neighboring cells can communicate with each other. They don't talk, but communication here means transferring of substances. Okay. Is it clear up till now?
Yes.
Any doubts?
No.
Students, you can show a little bit of enthusiasm. Please, please don't make it an atomic gathering class. Please, I really, really don't want it to be an atomic. All right. So talking about the second phase, this is the elongation phase or phase of enlargement. See, let's do one thing. When I say, did you understand? So you say, yay, okay. So that I'll know that even if it is fake enthusiasm, still enthusiasm is enthusiasm, okay?
Okay.
So did you understand?
Yay.
All right. So let us talk about the second phase of the growth. So second phase is the elongation phase, or you can call it as the phase of enlargement. Now, this phase comes right next to the phase of meristematic, means the meristematic phase. So right next to the meristematic phase is the elongation phase. Now let us see what are the qualities which are found in the cells which are present in the elongation phase. Like how we saw for this thing, meristematic phase, same way we'll see now for the elongation phase. So here the first quality which is there in the cells of the this thing, elongation phase is increased vacuolation, meaning the number and the size of vacuoles would increase according to the growth. So increased vacuolation means number and size of vacuoles are increasing. The second quality which is present is the new cell wall deposition. Now, cell wall was primary in the meristematic phase. Now, slowly, new materials will start getting deposited on the cell wall so as to modify it into a secondary wall maybe in the later stages. So that is why cell wall deposition is observed during the elongation phase. This is the second phase. Then the next characteristic found in the cells of the elongation phase is the cell size. Now the size of the cells would increase. Why? First of all, because vacuolation was happening, vacuoles were increasing. So if this is a cell and this is a vacuole, so if I say that the vacuole has been increased like this, so wouldn't the cell also increase inside?
Yeah.
So this is the reason why the cell size also increased with like as a proportionality to vacuolation increase. Is it clear up till now?
Yes. Yay.
Sure?
Yay.
Okay. Now coming on to the third phase, that is the maturation phase, or you can call it as the phase of differentiation. Maturation phase or the phase of differentiation. Now, the cells which are present next to, like how next to the meristematic phase was the elongation phase, same way next to the elongation phase is the maturation phase, or you can call it as the phase of differentiation. Now the cells of this zone, they attain their maximum size and they have thickened walls and modification in the protoplasm. Now those cells are being matured slowly and slowly. So we'll have modifications. We'll see and observe the different modifications. And what are those? First of all, the cell has reached maximum size. Then the cell walls, since depositions were being done to it, more and more deposition caused the thickening of the cell wall. Now the cell wall can be considered as secondary cell wall. And then also the protoplasm got modified. So these are the following things or the characters of the cells that we observe in the maturation phase. So are the three phases clear to you?
Yeah.
Meristematic, elongation, maturation. Any doubts?
No. No.
Okay. Now you people can note it down.
You like this chapter?
You're asking me whether I like this chapter.
Mm-hmm.
This chapter gets interesting in the next topic. Right now it's just the introductory part. So it seems boring.
Right now it just looks like anatomy.
Really? I find it easier than anatomy. Aren't you finding it easier than anatomy?
Yeah. But then it's like still. So
Hmm.
similar.
See, slowly, slowly it'll get better. It's not as bad. Once we get onto the phytohormones, meaning the plant hormones, then it is very interesting. I mean, as far as I think, let's see what you people will think.
Done.
Everyone's done?
Yes. Done.
Everyone's done.
Yes,
Hmm?
ma'am. Yes. Done. 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 Done, ma'am. Done, ma'am. Done, ma'am.
Both of you are done?
Yes, ma'am. Done.
Done?
Yeah. Done. Done.
Okay. So now let us talk about the growth rate. Now, what is growth rate? So the increase in the growth per unit time, always remember, per unit time is defined as the growth rate, meaning how much growth is happening per unit time is the growth rate. Now, growth rate can be expressed in many ways, in fact, but here we are going to see the two ways by which the growth rates can be expressed. The first one is arithmetic, the second one is geometrical. Let us talk about the first one, that is arithmetic growth rate or arithmetic growth. Now, it is the growth rate. Basically, here a constant rate is at which the growth is occurring from the beginning and up till the end. Okay, and it progresses arithmetically. Now, what happens here is that after the process of mitotic division, only one daughter cell would divide continuously, whereas the others would undergo the differentiation and become mature. So the division aspect will be only for one daughter cell. The rest of them would differentiate and start becoming mature. Okay. This is what happens according to the arithmetic growth. The equation by which we can express the arithmetic growth is LT is equal to L naught or L0 plus RT, in which the LT stands for the length of the organ at the time T, whatever time we have given. L0 stands for the length of the organ at the time 0. And then the R stands for the growth rate or the elongation per unit time. And when we do so, a linear curve is obtained. Linear curve means this one. A linear curve is obtained. Okay. And if we consider the two axes, so the first one, if we are taking example, is the height of plant, and here we are taking time as the x scale. Okay, so this is a linear curve which is observed in the case of arithmetic growth. Any doubts up till now? All right, now moving forward, let us talk about the geometrical growth. Yeah.
The arithmetic thing, I didn't exactly get it.
See, it is the pattern of growth that they are trying to make us understand, wherein one cell would divide, okay, and then what will happen is that the other cell, okay, the other cells, see, after the mitosis is done, two daughter cells are formed, isn't it? One of those two daughter cells would divide continuously. The other daughter cells would undergo maturation. They would mature. Okay, and for this only, they have given an equation. This is the equation, and the curve obtained is a linear curve. Got it? Any doubts?
No.
All right, now moving forward, let us see what is a geometrical growth. Now, geometrical growth is the growth where all the cells would be dividing, meaning after mitosis, like whatever cells are formed, 2, 4, 8, all the cells would divide, and the daughter cells will grow and divide yet again. So if one cell is dividing and making two daughter cells, then each of these two daughter cells would also undergo division. This is how the geometrical growth will take place. Okay.
Okay.
Now, in the geometrical growth, there are phases that are seen. Lag phase, log phase, and then there is a stationary phase. So talking about the first phase, that is the lag phase. So this is the initial phase of the growth, meaning at this point, lagging, you understand? The slow growth rate is obtained here in the lag phase. Then comes the log phase or the exponential phase. Here what happens? Rapid increasing of the growth is done. And then at the third stage, we have the stationary phase. Stationary phase. Now let us see what happens over here. So during the stationary phase is like stationary, it stops. Okay, meaning it almost goes to the zero. So the growth stops. Okay, not exactly you can say limited, because of the limited nutrient supply, it leads to a stationary phase. Okay, because of limited nutrient supply. Now, basically, like how arithmetic equation was given to explain the arithmetic growth. Same way, the equation which is given to explain the geometrical growth is as such. We have W1 is equal to W0 E, sorry, E or T. This is the equation. You people have to just remember the equation. No numerical will come up. Just remember the equation. W1 is the final size. It is the final size.
Then why
Now
we
this size,
learning the equation?
because they can ask you what is the equation for exponential or arithmetic growth. They will not give you numericals on this. Okay. But you should know. Like, since it is, you are learning because it is given in the NCERT. And now talking about W0, it is the initial size when it is beginning. Okay, so initial size, and then obviously R would be the growth rate, and then T is the time of growth, and lastly we have E. It is the base of logarithm. Do you people solve logs in physics? Hmm?
I guess.
I guess you people haven't came around to that.
I mean, it's rare
Okay.
sometimes.
And this R can also be known as the efficiency index. Okay, meaning how efficient the plant growth is, that is measured with R. Okay, so efficiency index is the R. Okay, now if you were to plot out,
R is also growth rate.
same thing, you can write
It's the
here
same
only,
thing.
or efficiency index.
Oh,
Okay, why? Because as it measures the ability of the plant to produce new plant material, okay, so this is why we also call it as the efficiency index. Okay, now if we were to plot a graph for this particular growth, so we'll do this like how we normally plot a graph, only straight. Then here you'll write size slash weight. Hmm. I'm writing what you'll write in straight. Okay, size slash weight of the organ. It will be taken as the y-axis, and here we'll take time in the x-axis. Now if you were to find out the curve, so it goes like this. This is the lag phase where growth is slow. Then this is the log phase or the exponential phase where the growth is very increasing exponentially. And then we come to a halt, that is stationary phase. It happens when the resources are limited. Now, what is the shape the curve is? Here the curve is what? What type of curve is this? It is a sigmoid curve, okay? A sigmoid curve is obtained. Also, with respect to this particular sigmoid curve, we can also say that a sigmoid curve, this is a characteristic feature of living organism growing in a natural environment. So it is more ideal. Exponential growth is much more ideal as compared to the arithmetic growth. Okay. So we can say that a sigmoid curve is the characteristic of living organism growing in a natural environment. Got it? This much is clear. So if somebody asks you, a question can come like, out of the two growth equations or the two growth models, which one is more this thing? Do you call it practical? What would your answer be? Hmm?
Can you repeat?
The question is, which one of the two curves obtained is practical?
Sigmoid.
Sigmoid curve. Okay. And we can say which growth is more practical, exponential or arithmetic?
Exponential.
Exponential. Now, see, growth can further be of two more types. Or you can say the growth rate can also be of two more types. The first one is absolute. Absolute growth rate. And then we have the relative growth rate. Relative growth rate. Now, talking about the absolute growth rate, see, basically, when you compare the total growth per unit time, then that is your absolute growth rate. But then, if you relate it with something, then that becomes the relative growth rate. For example, if you relate it with the initial, initial growth or initial size, then that becomes a relative growth rate. Okay. And if you turn on to page 244 in your NCRT, look at the figure 15.7.
Yes.
Okay. Now,
Mm.
look at the first leaves diagram. Now, amongst in both of them, which one do you think would have a greater growth?
Second one.
Second or the first one?
Can you repeat?
Which one would have greater growth rate?
A.
The A one, right? Why? Because see,
Because it's
initially it
still
was
small.
5. What?
Ring.
It is still small. Yeah, it is still small. But we are talking about the way the growth is happening in them. So if we were to take a talk about the smaller leaf, so initially it was 5 centimeters. Then it grew to 10 centimeters. So we take the difference. And then we multiply it with 100. What do we get? 100%. Okay. Again, of no more particular importance, no significance. Up till now, is everything clear?
For the second one, how do we do it?
The second one is like, what is the amount of growth occurring? 5. What was the initial? 50. Multiply it with 100. What do we get? We get 5 to the 10% growth. Correct?
Oh.
That is why. It doesn't matter on the size. It matters on the rate of the growth. Got it? Got it?
Okay.
Yes. Now you can note it down.
No. Done. 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 Done.
Did you just unmute and sigh?
No, I said done.
Oh, I thought it was like unmuted.
No. Why are there two log faces?
One is lag, one is log. The upper one is lag, LAG. The lower one is LOG.
Lemma. Done. Done.
Done, ma'am.
Can you scroll a bit up? No, down. Done. Done.
Yes, ma'am.
Done.
All right. Now let us see what are the certain conditions for
What
growth.
is the difference between
Hmm?
we already did growth rate, right? Growth rate was arithmetic and geometric, and then what is absolute and relative?
See, the total growth rate per unit time. Total growth rate per unit time is absolute growth rate. Okay. Whereas the like depending upon the initial parameter, if the growth rate depends upon the initial parameter, then that is relative growth rate.
What is the purpose of all this?
Nothing. Because this whole chapter is about growth only, right? That is why we are seeing growth rate and everything.
Yeah.
So the growth rate depending upon the initial parameters. When you relate it with something, that is growth, a relative growth rate. But when you see it per unit time, then that is absolute growth rate. Now let us see what are the conditions that are required for the growth. So water, oxygen, then nutrients, then the right temperature. Apart from that, we have optimum temperature is done. Okay, and under the nutrients, we have macro and micronutrients. The ones that are needed in a larger amount is macro. The ones that are needed in a smaller amount is micronutrients. So both of them are required. And the last and the most important one is turgidity of the cells. Turgidity of cells. Now see, turgidity of cell, what do is that they help in the extension of growth. Help in extension growth. Okay, meaning they help the cells to extend the turgidity, the temperature, the nutrients, the oxygen, the water. These are the certain conditions that are like absolutely necessary for growing in terms of like in the case of plants. Now, there are three terms that are so confusing. Okay, that you will get most confused here only, or maybe not. So we have differentiation, one term. Okay, then we have de-differentiation, de-differentiation, and lastly we have redifferentiation. These are the three terms that we need to talk about, and we need to find the difference in between them and what do they mean. So differentiation, de-differentiation, and redifferentiation. Now, look, the cells from the root apex or the shoot apex, or you can say the apical meristem, they would divide, isn't it? And the cells from the cambium, they would also divide, and then they would mature in order to perform certain functions, right?
Yes.
This whole thing wherein a cell matures, okay, this whole thing meaning when they will divide and then they will mature, this thing is known as differentiation, normal, okay? So talking about the first one, the first term is differentiation. Differentiation means the division of the cell, the division of the cells, and then the maturation of the cells, maturation of the cells in order in order to what?
Divide.
No, in order to perform specific functions. Why do they get matured? So that they can perform specific functions. Okay, so this whole process is known as differentiation. Got it?
Yes.
Hmm. Now what happens is that sometimes now when the cells have become matured, do you remember permanent cells, permanent tissues? Now they don't divide anymore, right?
Yes.
So what happens is that they become permanent. But then plants have this interesting phenomenon wherein the cells that have lost the capacity to divide, they can regain the capacity to divide and then divide again. And this term is de-differentiation, meaning the phenomena by which the cells that have lost the capacity to divide can regain the capacity of division under certain condition is known as de-differentiation. Okay? So the second one is de-differentiation. In this case, what do we say? That it is the phenomena where the cells that have lost the ability lost the ability to divide regain the division capacity under certain conditions. Okay, there are some conditions during which this can take place. So under certain conditions, so we call this phenomena as the de-differentiation. Okay. So we'll also put examples over here, like how in the case of differentiation, we had the example of root and shoot apical meristem. Here we can put the example apical meristem, whether it is in the case of root apical meristem and shoot apical. But talking about the de-differentiation, so we can give the examples such as interfascicular meristem or interfascicular cambium. Interfascicular cambium is one such example, and cork cambium is another example, meaning these would again get the division capacity, meaning the formation of interfascicular cambium and cork cambium from fully matured parenchyma cells. Parenchyma cells. So when these cells regain their capacity, they form the interfascicular cambium and cork cambium. Okay? You can, before that, you can write down formation of interfascicular and cork cambium from the fully matured parenchyma cell. Okay? Now,
I don't understand the first one.
which one?
Differentiation.
Differentiation is a normal term wherein the cells would divide and then they would mature. Why do they mature? So that they can perform a specific function. This process of division and then maturation is differentiation. Now what happens is that when a cell becomes mature, then they lose the ability to divide. So another interesting phenomena is de-differentiation, wherein the cells that have lost the ability to divide can now regain the capacity to divide. And the example over here is the formation of interfascicular cambium and cork cambium from the fully matured parenchyma cells. See, parenchyma cells became fully matured, they lost their ability to divide. But then they regained that ability and they made the interfascicular cambium and the cork cambium. This is de-differentiation. Got it?
Yes.
Now, now once they have been de-differentiated, they can again lose the capacity to divide, okay, and they perform a specific function. So now it means that they have gotten redifferentiated, meaning now again they have lost the capacity to divide. Okay. So now we can write down redifferentiation. Here what happens now? The cells, or we can say the meristems or tissues, okay, that are able to divide and produce the cells that once again lose the capacity to divide. We can say cells when they once again lose the capacity to divide are termed as redifferentiated. This is the phenomenon. Okay. Any doubts?
No.
Any hatred?
Yes.
All right. Now you can note it down.
I wrote it.
What about the others? Khadija, Khudaija, have you people noted it down or do you need to note it down?
We have noted now.
Noted?
Yeah.
Okay, good. Now moving forward, let us talk about development. Now development, all sorts of development happens, right? In the tissues, in the organ, in all the different body parts of the plant, development takes place, right? Now, see, there are two phenomena that we can actually discuss over here is plasticity. Oh, not actually, this is a great phenomena. This is cool. Talking about plasticity. Now let's see what it says. Now see, plasticity is like one plant would follow different pathway in response to the environment to form different structures. This ability is called as plasticity. Meaning, for example, in one leaf, for example, if you look at the leaf of a coriander, so it is all sorts of shape in one only. So we can say that they have actually the property of plasticity. Okay, this is known as heterophily. Heterophily means different types of leaves. So when we talk about the heterophily, we say that this is known as plasticity. Meaning, when you look at the leaves of the coriander when it is young, it would look completely different. And when you will look at the leaves when it is like matured, so then it will look completely different. This property is known as heterophily, okay, or plasticity. Got it?
Yes.
So for now, for today's class, let's just write the example of plasticity. So we'd say heterophily. Heterophily means different types of leaf. So heterophily in which plant? So it occurs in coriander. It also takes place in cotton. And there is another plant that is known as larkspur. It takes place here also. Got it?
Yes.
So that is, let's just keep it till here only for today's class. And the next class, hopefully, let me see what is the next thing that will be starting in the next class. Hopefully it is interesting so that my students don't run away.
Plant
Yeah,
growth.